We move on to the next session that's on new emerging technologies. May I now invite the chairs for the session, Dr. Kelly Bateman and Dr. Vindhya Mohindra, onto the dais, please. I also welcome the session coordinators, Dr. G.K. Shivaraman and Dr. Purnima. In this session, we have two presentations, a keynote presentation by Dr. Argus Sunato and a lead presentation by Dr. Eduardo Liano. May I invite the presenters also onto the dais, please?
Good afternoon to August gathering. It's my pleasure to introduce Dr. Kelly Bateman, who is a renowned pathologist. And she is invertebrate pathologist at the Center for Environment, Fisheries and Aquaculture Science, CFOS in UK. Her work focuses on aquatic pathogens and combined approaches based upon histopathology, transmission electron microscopy, and molecular systems for the classification of novel and emerging pathogens, in particular disease of crustaceans. Kelly is leading the work of crustacean health team within the WOAH Collaborating Center for Emerging Aquatic Animal Diseases and the International Center of Excellence for Aquatic Animal Health at CFOS. She has more than 20 years of experience in the diagnosis of diseases in experimental, farmed, and wild aquatic animals, with special interest in the identification and characterization of novel and emerging pathogens of crustaceans. Kelly is an author for more than 50 peer-reviewed publications in the field of aquatic animal health. Now I request Dr. Shivaraman to introduce Dr. Vindhya Madam.
It's my great pleasure to introduce the chairpersons, Dr. Vindhya Mohindra. Dr. Vindhya Mohindra is former principal scientist and head, Genomics and Computational Resource Division, ICR, National Bureau of Fish Genetic Resources, Lucknow, India. Dr. Vindhya was also coordinator of ICR Consortium Research Platform on Genomics for 10 years. And Dr. Vindhya has been conferred M.S. Swaminathan Award for Best Indian Fisheries Scientist. Her research interests include aquaculture genomics and conservation genetics in natural populations of fishes. Dr. Vindhya has handled more than 11 externally funded research projects and guided 11 PhD students and 2 postdoctoral research students. And she is also academic editor for more than 4 prestigious journals. In the course of over 30 years of her research experience, she developed genetic and genomic resources, including whole genome assemblies, transcriptomics, and trait-associated genes and molecular markers. Additionally, Dr. Vindhya has also studied signatures of natural selection and genomic diversity in important freshwater fish species to build genomic resources and to gain knowledge about functional biodiversity. At present, Dr. Vindhya has over 150 publications. Thank you.
Good afternoon. Welcome to the technical session 6th on new emerging technologies. Nowadays, different technologies are being developed for the aquatic animal health management regarding the molecular mechanisms for the host-parasite interactions, novel RNAi vaccines, vaccine target discovery, and many, many areas. For this session, we have 2 invited speakers. Keynote presentation is by Dr. Agus Sunato on genome engineering in aquaculture from genetic modification to gene editing. Second speaker will be Dr. Liano. He has a lead presentation on innovative technologies on alternatives to the antimicrobial, towards mitigation of antimicrobial resistance in aquaculture.
Is this working? It's my great pleasure to introduce our first speaker. So Dr. August Sonato is Team Leader of Aquaculture Biotechnology at CSIRO in Australia and a distinguished molecular virologist with a PhD from the University of Queensland. Dr. Sunato's groundbreaking discovery of the Indonesian KHV C07 isolate laid the foundation for over 15 years of research into viral biocontrol of invasive carp, contributing directly to Australia's National Carp Control Plan. Before joining CSIRO in 2013, he led the Fish Health Research Laboratory under Indonesia's Ministry of Fisheries. His work now integrates viral and genetic biocontrol technologies, advancing sustainable aquaculture and precision genome engineering. Dr. Sonato's leadership in tilapia biocontrol, gene editing, and aquatic biotechnology continues to shape global strategies for resilient aquaculture and invasive species management. And I hand the floor over.
Okay, thank you. Thank you, Kelly, for your very warm introduction. I appreciate it. Thank you again, Dr. Kuldeep and India team for organizing DAA12 in this beautiful Chennai. I managed to sneak out this morning and visited the fisherman village, which is really live and exciting. So let's try if the technology works. It does. So I will start my talk by clarifying the differences between genetic modification using recombinant DNA technology and more modern gene editing using CRISPR-Cas9. This distinction is important to understand both the science and the regulation landscape. So I will then highlight some of the application of gene editing, for growth rate, disease resilient, climate change, the usual suspect. Of course, this technology not without challenges. So I will share the challenges in the lab, but also try to identify what are the challenges that may be facing in the future. At the moment, for example, during often the workflow of our gene editing, we identify two bottlenecks. One is how are we going to find the right gene target for specific trait? And then second, how we develop more efficient, high throughput delivery. And then I will finally touch a bit about the barrier for adoption. As any new technology, there are barriers to adoption. I will not discuss all the ethical consideration, the social license, and so on, but I will probably focus a bit on the government regulation. Let's see. So let's start by looking at genetic modification, GM. This technology discovered in 1972. Some or most of you hasn't born yet, so it's pretty old technology. So people use DNA recombinant DNA technology to insert foreign DNA from one species into another species, which cannot be achieved by traditional breeding. And because there is foreign DNA, new DNA in the host, the resulting product was so-called genetically modified organism, GMO. The well-known example is aqua advanced salmons, being the first transgenic GMO animal, all right? Animal, not only fish, has been approved by FDA for human consumption. But it took 22 years, cost 20 million to grab approval. In contrast, gene editing, for example, using CRISPR-Cas9, discovered in 2012, allow us to precisely change small nucleotide sequence of the host genome without integration of foreign DNA, which could occur naturally. For example, if you like sunbathing, your skin is exposed to UV. The UV damage your DNA. And then during the DNA repair mechanism, mutation happen. And that's the same. We cut the DNA not using UV light, but we cut the DNA using nuclease, enzyme, CRISPR-Cas9, right? Because there is no foreign DNA inserted into the genome, it is not considered as a GMO in some country. An example in Japan, there are already 3 gene-edited fish, sea bream, tiger puffer, olive flounder, considered as safe and commercially available in Japan. So if you go to Japan, you can actually order online. I went there in October last year, but you cannot order it from outside Japan. There are gene-edited tilapia are being farmed in Brazil as well. So how scientists create this fast-growing Atlantic salmon? So they cut genes, growth hormone genes from Chinese salmon, and antifreeze promoter from ocean food, so that they can active throughout the year, not only during summer, but also during winter, because the antifreeze promoter. Then put these 2 genes from 2 different species into Atlantic salmon, resulting in the genetically modified salmon, fast-growing salmon. And as I said, it took 20 years, $20 million to get approval. Gene-edited fish, however, only take few years to produce and a couple months to get approval. You can find information of gene-edited animal, including fish, in this database. So you can search by species, country, trait, gene target, and so on. For example, here, if you check by species, there are 4 fish species, tilapia, red sea bream, tiger puffer, and olive flounder. And there are only 2 other species, cattle and pig. So gene editing in aquaculture is in the front line of gene editing animal. And then you can see here that in 2001, 2003, 21, 22, 23, there are 3 gene edited is registered. So it's pretty quick process. It's not surprised that the target is growth rate. So, but different gene target. For example, the red sea bream, they're targeting myostatin genes with the muscle regulator. But for the olive flounder, they're targeting leptin receptor. Increase the appetite, grow faster. Easy. So now, what is CRISPR gene editing? So CRISPR-Cas9 consists of 2 main components. One is CRISPR-Cas or CRISPR-associated nuclease, we call it Cas. It's an enzyme that cuts DNA. And another component is guide RNA that direct nuclease to cut DNA at a specific location. It's like molecular scissor. The scissor is the Cas9, the enzyme, and the hand is the guide, who direct the enzyme on the scissor to cut DNA in targeted area in a specific location. Now, in the CRISPR mechanism, there are 2 mechanisms. One is what we call non-homologous enjoining. So when CRISPR-Cas cut the DNA, double-strand break, our system using DNA repair mechanism will join the damaged DNA. During this process, mutation happen, deletion, insertion, and so on. And that is ideal for gene knockout because insertion and deletion will disrupt the gene function. And that's what categories as site-directed nucleases. This is no nuclease directed to particular site. The resulting product is non-GMO because there is no template, there is no foreign DNA inserted into the genome. The homology-directed repair is different. As the name suggested, this is repair using homology sequence from other species, the foreign DNA basically. So when the nucleases cut the DNA, we introduce homology sequence into that space. That will create gene knock-in. So whatever gene you want to be, growth, disease resistant if you have it, and so on. So basically, you have gene target, cut it from other species, or create it synthetically in the lab, and then put it in the genome. And that's what is considered as, or categorized as SDN2 and 3. And because there is foreign DNA in the genome, the resulting product are considered as a GMO. I'll just give you an example of regulation of this technology in Australia. As I mentioned earlier, there are 3 classifications of gene editing technology that relevant to the regulatory consideration. So one, well, first is SDN1. There's no template, not a GMO. SDN2, you use short DNA template. It's considered as a GMO, but not as strict as SDN3, where you use long DNA template. So for aquaculture purpose, we want to develop product that is not GMO. That's why in our lab, for this reason in our lab, we use DNA-free gene editing. So we are not using DNA plasmid, we are not using messenger RNA, but we are using protein, all right? And that is for gene knockout only. So we are not doing knock-in, but knockout. And thereby avoiding the GMO classification. Looks good, we have the technology, but before you jump into the lab, edit any phase gene editing, please check the regulatory in your country, right? Because regulation of gene editing in country is different. Even in Australia, regulation of gene editing in the mainland and in Tasmania, where I base, is different. So we are engaging, for example, with the Department of Tasmania, who authority for the gene editing stuff, to consider SDN1 organism as a non-GMO, because currently, although the federal government rule is it is not GMO, but Tasmania considered as a green, no GMO, so there is, but the good thing is they are engaging with us, right? This is, what do you think, guys? So then we sit down together and then we respond to the question why SDN1 need to be considered not as a GMO. Anyway, bottom line is check with the federal government, it could be Department of Health, it could be Department of Agriculture, Fisheries and Forestry in Australia, your state government, the authority, gene technology regulator in Australia, and then food safety, Australia, New Zealand, and your institutional biosafety committee in your institute. And then you start thinking, all right, what lab that we need to build? So again, check the regulation because they probably need at least 2. It is physical containment. In our case in Australia, we need to talk to OGTR, Office of Gene Technology Regulator, which is under Department of Health. But the biological containment, we talk with Department of Agriculture. So different department working on different area. And then permit, if you import Cas9, can you use, it could be used in vitro in the lab, but can you use it in vivo in fish? So check it with the regulation, and then animal ethic and so on. And then we have a lab, we have comply with the regulatory requirement. And then we start developing skill. The way we do it is we work with university. Because university, if they have faculty of medicine, most probably they have zebrafish facility, right? So zebrafish is not in aquaculture space, zebrafish also be used as an animal model in the medicine. So, and most probably they already have the capability of gene editing. So talk to them and then learn how they set up and so on. And that's why we start with zebrafish. And as again, we use as SDN1, and then we use standard transfection using microinjection of fertilized egg. So we have capability in zebrafish and in salmon now. Just to keep you in what we do in the lab in the first thing, as I mentioned earlier, there are two components of gene editing, Cas9, the enzyme, and guide RNA. So with Cas9 is just one of them. There are many different Cas systems. We tested, for example, Cas9 and Cas12. Again, we use ribonucleotide protein, which is protein, not plasmid, not messenger RNA. And here we find that Cas9, not surprising, is still better than other Cas system. And then you optimize the RNP and so on, pretty standard. And then testing different guide, just like you guys testing different primer pair. Super easy. So in here, for example, we use LSE45A2 with its pigmentation gene as a marker. So basically proof of concept that we can edit fish. EC1 is a pigmentation because we can see the color developing or not. So on the left side is the guide that doesn't work, and then right side is guide that work well, produce albino fish. Here we go. So and then we start analyzing the gene edit phase, genotyping, phenotyping, and so on. So on the right side now is Cas9 targeting ESO1, which doesn't work well. And the result is the face look patchy, still have some pigmentation, but not complete. But exon 6 works well, is completely albino. So yeah, testing different guide, testing different region of the genome, and so on. So now we have a good gene editing workflow, from gene target, design guide RNA, testing in vitro in cell culture system with GAP for sure as standard, and then micro-injection of single cells fertilized egg, genotyping, phenotyping, and then experiment with fish in the wet lab, to analyze the result of gene editing. So we're pretty good in that. But we also facing two bottlenecks. One is what is the right gene target for particular trait? If we are looking for disease resistant because we are at DAA, so what the target, what the gene target? And then second is microinjection is beautiful, it's work, but you need to inject individual egg. You can imagine how many hundred or thousand you need to inject. Question is, can we use sperm, for example, as a delivery mechanism for gene edit? Sperm deliver information from us. Can we use it to deliver the cargo? So that two bottlenecks that we are facing, and we are working on it. Gene target identification. So where we are interested on disease relation, considering my background. So we started to work to ask question: is there any immune gene that we can target it to improve immune resilience in fish? So we started to knock out individual immune gene. For example, here REC1, which is play in the antibody production, the VDJ recombination and T-cell receptor. So REC1 knockout should have no adaptive immune system, right? So we start working on that area. And also toll-like receptor, TLR5. So TLR5, for example, recognize bacterial flagellin and stimulate cytokine. And it's unique in fish because they have two kind, the membrane bound and the secreted bound, and they have behave differently. So that picture is just to show the SLC45, which is the pigmentation gene, as an indigenous marker for the gene editing. Because when we edit immune gene, for example, we cannot say the phenotypic until we test them. So, but we would like to see quickly if the gene editing works. So we put SLC45A as a marker upon this process. Again, as I mentioned, we individually targeted each gene. Now, can we do more systematic approach using single cell RNA sequencing and spatial transcriptome? So we had Amanda and Joel working in this area. The disease may be not applicable in tropical, but the approach is applicable for any disease and any species. We are living in Tasmania, and by default we are facing this issue in summer. So in Australia, Atlantic salmon grown in Tasmania, where the water upper limit is, in summer could be 20 or more. So we have a bit of issue with summer mortality. We have been working with the breed for years, and studying the genomic of Atlantic salmon summer growth from 40,000 fish, 14 years to differentiate at Tasmania. They use genome-wide association studies, 50K SNP from 20,000 progeny for genotyping. And as you can see here, the results suggesting the polygenic nature of heat tolerance, meaning that, that heat tolerance is associated with multiple genes. So editing one gene may not have a big impact on the thermal tolerance. So that another challenge is working with the polygenic nature of a particular trait. But we are working on, in particular, with the one gene, S39 in the chromosome 28. Right. So the second bottleneck I'm going to talk to is delivery. As I mentioned earlier, microinjection is standard. It's well-established technique, but it's slow because we need to inject individual fish. It could be inefficient because causing mosaicism, so it requires breeding strategy to create pure gene-edited line. So we need a high-throughput delivery. So different team in a poultry gene editing, they developed a technology called sperm transfection-assisted gene editing. So use sperm to deliver the cargo. This is work in chicken. They enable generation of gene editing chicken in one generation. So we're testing it in fish, and ta-la-la, it doesn't work. So we compare microinjection, which is work beautifully. You see the wild type and the albino, and we sequence the DNA and we kind the indel in the gene-edited fish. The phenotypic is beautiful, all albino. We change the technology and the technique from microinjection with sperm. We can't find any edit, so it's not work. So technology that work in one species doesn't mean it work in another species. But we not give up yet. So, for example, can we use something to help sperm uptake? For example, can you use cell-penetrating peptide to facilitate sperm uptake of the CRISPR? So we tested 12 different CPP, and 3 of them looking good in terms of sperm uptake, based on the microscope. The viability is a bit issue. Some is pretty low, 10%, some 90%, and that's another level of challenges in the lab. On the last topic that we talk a bit is barrier to adoption. So I'm not going to talk about ethical consideration, socialization, public acceptance because of time concern, I guess. But I will talk a bit about regulation. So the global gene editing regulation tracker, you can find it in the internet, provide a good guideline what the regulation status, from prohibited score 0 to perfect 10, no unique regulation. I pick up just 2 there. No unique regulation means gene editing animals that do not incorporate DNA from another species are regulated as a conventional animal with no additional regulation. And then lightly regulated is some or all type of gene editing. So some is, for example, SDN1, but not SDN2 and 3. Regulated more strictly than conventional breeding, but not as strict as transgenic DMO. So as I mentioned, the regulation of gene-edited fish vary significantly between countries. Gene-edited is lightly regulated in Japan and Australia, the green color, but highly regulated in New Zealand. SDN1 product is not GMO in Australia, but GMO in New Zealand. So check the regulation in your country before you're doing any gene editing. I understand the regulations are under development in Asia and in Africa, and we are in the process of organizing workshop for gene editing, try to understand what the regulation landscape in the Asia Pacific region and what the technology and they are using and so on. So if you are interested on gene editing and aquaculture, whether from regulation side or from the technical side, please let me know. Just to summarize what I've been talking today, is we are moving from genetic modification, GMO, to gene editing technology, CRISPR, which could be not GMO. I also highlight the application of the technology, growth rate, disease resilience, climate adaptation, you name it. I also share the bottleneck. As in any new technology, it doesn't work straight away. One of my supervisors told me when I did my PhD, if your PCR work in the first place, you will learn nothing. So that's the same with gene editing. It looks simple, but it may not work in the first place. So be patient with that. And then I said the bottleneck, finding the right gene target and delivering CRISPR region in a high throughput system. Then I touched a bit about the barrier to adoption and, working to have a bit of workshop in the future. And with that, I think, thank you again for your attention. Thank you.
Thank you for a very interesting presentation. I think we have time for a couple of questions. So does anybody have any questions?
Very thankful for your presentation. It was a very nice one. Just wanted to ask whether DNA methylation has any effect on the efficiency of CRISPR technology?
Sorry if I get it right.
Does the DNA methylation does it have any effect on the efficiency of this technology? Because that's a kind of repair mechanism.
Yes, so we are not looking for the methylation. We are working as long as, so the efficiency of the technology is rely on whether you have a good target of gene for particular trait and whether that gene is a single copy. If you have multiple gene or in multiple copy, then you are in trouble. So looking for the right gene target for a particular trait that just associated with single gene or single gene copy is more important than the methylation.
Thank you.
Agus, great work.
Thank you.
Good start. Simple question. How do we use the technology for mass production of fry and seed, which we need in billions for the next 10, 15 years?
So application of the technology to mass production of the fry. So I guess what we are thinking is we edit single cell fertilized egg, so that when the cell multiplying, all the cell containing the same gene edit. And then we use that as a broodstock. And then we use broodstock, to produce the fry. Isn't that?
Which one? You will edit the sterile ones or non-sterile? Will you edit the sterile fish or non-sterile fish?
Very good question. So yeah, I'm not touching about the sterility yet, but you are asking the very fundamental question, because 2 points. One is we use sterile fish to protect IP, if you are working with company, for example. And second is to protect the environment, because we don't want the gene-edited fish made with wild type. So we use sterility to protect that. How we are going to do it? We have some ideas, but we haven't get there. But the bottom line is we work with the breeding team how to manage the broodstock.
Thank you.
Thank you for a really educating talk as far as I'm concerned. Epidemiologists moved away, evolved from Koch's postulates of disease causation to causal webs, and Evans was one of the people involved in that. And for me, access to the genome just gives us access to another range of risk factors, like age, sex, food, temperature. Now we have genes to consider, and we throw them in there. And you would expect it to be multifactorial. You would not expect to find single gene associations, as you've shown here. But also, there's a precociousness amongst proteins, in that if you actually remove one protein, there are others which may not be major players, but still have the capacity to take over a function. So I think for an epidemiologist, it's great to have this resource, and the more we have it, the more we can put into multivariable analysis associated with the disease. So,
I cannot agree more with you, Kenton. So yeah, really good. And we completely agree with that interaction. Thank you.
I think we have one more question.
Hi, I'm just going to add just one comment to Dr. Kenton's comments here. What are we doing with the plasticity of the fish?
Good question. My understanding of breeding genetic heritability is 101. Very good question, but I don't think I'm in a position to answer your question. So my background is coherpes virus. Yeah, not breeding, sorry.
Maybe a common sense question, like the gene edited ones will always breed through?
Sorry, Dr. Sangkar, come again?
Will the gene edited individuals will always breed through? The next generation will pass on?
Oh, yeah. So if you edit the broodstock, the offspring will, because the genetic makeup will be the same. Because the genome isn't the same. It is heritable now.
Okay, thank you. I'd like to thank our first speaker once more. Thank you.
Thank you.
Thank you so much. Our next speaker is Dr. Eduardo M. Lino. He is speaking on innovative technologies on alternatives to antimicrobials towards mitigation of antimicrobial resistance in aquaculture. He is Director General since 2024 to present, and Senior Program Officer since 2010 of the network of aquaculture centers in Asia Pacific, that is NACA, based in Bangkok, Thailand. He oversees NACA's regional programs, manages a secretariat, and leads initiative on health and biosecurity and food security, safety and certification. He provides expert guidance on aquatic animal health, supporting Asia-Pacific countries in disease surveillance, antimicrobial resistance, and biosecurity. Dr. Linio serves as Secretary to the Asia Regional Advisory Group on Aquatic Animal Health and Secretary and Treasurer of the Fish Health Section of the Asian Fisheries Society. He is also active in several WOAH advisory groups and AP Aqua Net Steering Committee. Previously, he was a researcher at Aquaculture Department of Southeast Asian Fisheries Development Center and held postdoctoral roles at Taiwan Fisheries Research Institute and National Taiwan Ocean University from 2002 to 2010. Dr. Linio has authored over 60 scientific publications and co-edited 8 books in aquaculture and aquatic animal health. Dr. Linio, please.
Thank you, Chair, for that introduction. So I think this is just my second time to present a paper in DAA. My first was in DAA in Phuket, I think DAA12. Ever since, I'm just attending as a chair or as a moderator or something. So this is a special DAA for me, as I was invited to have a lead presentation on innovative technologies on alternatives to antimicrobials towards mitigation of antimicrobial resistance. To begin with, just a brief, many of you already know this, that Asia Pacific is still at the forefront of aquaculture production. So we are producing around 90% of the world's production. However, its further growth is still essential. So to enhance productivity, a more intensive approach is essential, coupled with efforts to address ongoing and new challenges to maintain aquaculture sustainability. So despite of the development that we had in the aquaculture industry, the aquaculture sector still faces a lot of challenges and issues, including environmental pressures, climate change, shifting globalization, and of course, disease outbreaks. Therefore, business as usual is no longer viable for the aquaculture industry in the Asia-Pacific region. We need innovations and investment to transform aquaculture, which is needed for a major shift towards a more efficient, inclusive, resilient, and sustainable aquatic food system. So in the past few years, NACA, in collaboration with FAO, has developed this white paper on aquaculture transformation in the Asia-Pacific region in support to the Blue Transformation Roadmap of FAO. So still, aquaculture is a highly significant food production sector in the region, as it provides livelihood opportunities, especially for small-scale farming communities, which are common, very, very common in the region. And definitely it contributes to food security, nutrition, and poverty alleviation. As I said, aquatic animal diseases is still a bottleneck in the sustainability of aquaculture, which includes both important and emerging diseases. So the spread of these diseases, what we call it transboundary diseases, is a direct result or directly correlated with the transboundary movement of live aquatic animals and aquatic animal products. This transboundary movement is a necessity for development of aquaculture, so we could have not achieved what we had now in the aquaculture industry without transboundary movement. On the other hand, antimicrobial resistance, or AMR, is now a significant global challenge where important bacterial pathogens develop resistance to antibiotics, posing threats to aquatic animals, to human health, and to the environment. AMR is driven by a widespread and often prophylactic use or misuse of antimicrobials. And through this way of using antimicrobials, we are creating a selective environment for resistant bacteria to thrive and share resistant genes to other pathogens or bacteria in the environment. So the risk associated with AMR includes its risk on farm production, though it will have a higher cost, though antibiotics are not cheap, lower survival because of the diseases that we have, and failed treatments, because once we develop AMR, then most of the antibiotics will not work. Public health, the transfer of resistant genes to important human pathogens, which can create big problems once these resistant pathogens of human infect a wider human population. For the market and trade, so we have export restrictions and rejections of shipments due to antibiotic residues. And of course, now people are more concerned on the food safety issues of the different seafood commodities that they are eating. Also, a risk in food security. The AMR pose a long-term risk to sustainability of the aquaculture. So we often tell farmers, don't use antibiotics, but what alternatives can we offer? So without offering them alternatives, then we cannot just tell them, stop using antibiotics. So there are quite a few, several alternatives to antimicrobials, including vaccines. So you have heard a lot of presentations on this. Immune stimulants and phytochemicals also, probiotics and prebiotics. We have biofloc technology, quorum quenching, antimicrobial peptides, bacteriophages, nanobubble technology, and of course, effective biosecurity, including the use of high health stocks, the SPF, SPR, and SPT. You can ask Victoria here on the details of this one, because these are her expertise. So for this, due to time limitation, I will just talk on two alternatives, the novel bacteriophages and the nanobubble technology. But before I present to you, I would like to acknowledge my a source of materials that I use on these succeeding slides. For the bacteriophages, I thank Dr. Kalaya from Biotech, Thailand, and Dr. Kitia from Unifas in Kasetsart University. And of course, for the nanobubble, we have Dong Ha. Thank you for providing those materials. So if you will have questions later that I cannot answer, Kalaya and Dong Ha will be here to the rescue. So first, the novel bacteriophages. So a bacteriophage is a type of a virus or a viral particle that infects bacteria. So the word bacteriophage literally means bacteria eater, because they destroy their host once they infect it. So what is the use of phages in aquaculture? So phages are now available that are effective against major bacterial pathogens of aquatic animals, including Streptococcus, Aeromonas, Edwardsiella, Flavobacterium, and Vibrios. So phage concoctions can now be applied by injection, oral immersion, and the benefits of these phages is targeted pathogen, residue-free, and microbiome safe. However, there are challenges on the application of these phages at the farm level, so including formulation stability, host immune response, and regulatory barriers. So I will talk more on regulatory barriers a bit later. So now, as what Kalaya mentioned in some of the discussion, they have now the fudge cocktails that are available to treat specific bacterial pathogens in aquatic animals. So Kalaya's project is called Shrimp Guard. So they are developing these Vibrio phages as an antibacterial agents and mixed or combined with immune enhancers, including E-lysozyme, beta-glucan, and a universal Vibrio antigens. So this is still at the research phase and not yet pilot tested at the farm level, but I think this is how they will present these fudge concoctions to the farmers. But there is one company, the Unifaz, which already produce fudge concoctions against Vibrio pathogens in shrimps. They call it Lice-V or Lice-Vibrio. So the Lice-V is fast. It has around 15 minutes precise targeting Vibrio species. So the concoction will be formulated depending on the target pathogens that you are, or the target Vibrio species that the farm is having. So it is an innovative and effective fudge cocktail. It's all natural, it's non-GMO, and it promotes good ecosystem of microbiome. So far, the targeted, some of the important Vibrio species that are being targeted are Parhemolyticus, this causative agent of append, Cholerae, Alginolyticus, Vulnificus, and Harveyii. So this is the dose recommendation, but dosage can be adjusted by customized application. So this is one of their trial at the farm level, intensive Penaeus vannamei grow out of Super PL in Thailand in August to September this year, wherein there are 700,000 shrimps per pond stocked. So at day 17 of culture, the shrimp are infected by Vibrio species before using the Lice-V. And at day 40, there is a 100% reduction Vibrio species after Lice-V application. And at day 40, size 72 shrimps per kilogram has been harvested. So you can see here, healthy shrimps in the pond. But there are challenges at the farm level adoption. So one is the limited awareness of the farmers, awareness and understanding on the fudge mechanisms to prevent diseases and its application at the farm level. Of course, the fudges are expensive to produce, and there is a market competition for the cheaper antibiotics which are readily available in the market. So there is a need for post-sale support, including customization of the fudge concoctions based on water quality and target pathogens. And one of the main challenges is the regulatory pathway. So in Thailand, although the Thai Food and Drug Administration confirmed that the product is not classified as a drug, its registration pathway depends on its intended use. So the Lice-V, they presented the product as a feed additive, so it is applied via feed. So it is considered to be regulated as an ordinary feed additive. However, if applied directly to the pond water, it may be classified as a hazardous substance, requiring different approval process under environmental or chemical regulations. And if you tell the farmers this is a virus, and farmers will panic. So they don't want, so they have to introduce this. We eat another term, bacteria eater, so that, farmers will understand that, this might not be another virus that can cause problems in the future, so like the other viral pathogens of shrimps and fish. So it is important to relay the message to the farmer in a word or in words that they can fully understand. So, and is one of the challenge in relaying this technology to the farm. So now, okay, 6 minutes. So now we go to nanobubble. I will not talk much because Ms. Vin already presented this a while ago in her presentation. So this is nanobubble. What I want to emphasize here is nanobubble can stay in the water column for a longer time compared to the ordinary bubble and the micro bubble, which cannot stay in the water column. So whatever you make use of the nanobubble, whether it's as a disinfectant or a delivery system for vaccine, it can surely have a longer exposure of the fish or shrimp that you are targeting to these compounds or disinfectants that you are applying. So nanobubble is the technology which can be used for different functions in the production of fish and shrimps. So there are now a lot of companies producing nanobubble machines, so those machines that produce nanobubbles, and the nanobubble water. It can be an oxygen nanobubble, an ozone nanobubble, and usually it contains around 10 to the 7 to 10 to the 9 nanobubbles per ml. So it's very small. It's a size of the virus. So application of nanobubbles in aquaculture. So if you see here the Snesko circle representing the host, the pathogen, the environment, which is the one needed for the development of the disease. So for the host, nanobubble can improve fish immunity, while for the pathogen, it can reduce the bacteria through disinfection mechanism. And in the environment, it can improve water quality, improve the dissolved oxygen, reduce organic loads, and reset microbial community in the water. So there's a lot of potential for this nanobubble in order to have this, what we call preventive strategies for avoiding disease outbreaks. So these are some of the farm-level adoption of nanobubble in Thailand and later in the Philippines. So this is a solar-powered recirculating aquaculture farm in Patumtani, Thailand. They are producing red tilapia. So as you can see there, they have the oxygen nanobubble machine that produces the nanobubble, which improve, mainly used for water quality management. So all of these, including the monitoring and control of water quality, are, they use AI or Internet of Things to control all these parameters in the RAS system. So they said that using the nanobubble, they don't have much problem on diseases, and they can have an increase in production. So this is the pond culture, Chokanan Farm in Chiang Rai, Thailand. So they produce red tilapia and snakehead, and operates a hatchery for red tilapia for fry and fingerling production, wherein this micro nanobubble generator is being used. So this ozone micro nanobubble water acts as highly effective disinfection agent, increasing dissolved oxygen levels, and reduces the biofilm formation, which really helped in managing or preventing any disease problem. This is the shrimp farm in Calatagan, Batangas in the Philippines. We call it Casa Cumbre Monte Shrimp Farm. This is, they are culturing Pinaeus vaname in ponds, supported by daily water exchange of around 30%, and they use the nanobubble ozone water treatment. Also, they have a pure oxygen generator as a backup, and strong monitoring protocols to maintain high survival and consistent yields. So they are producing around 20 tons per hectare of shrimps with, per crop per year, with 3.5 crops per year. So what they did is they installed this nanobubble ozone generator in the supply canal. So the water from the sea will pass through this system, and it will then be deposited in the pond reservoir, the water reservoir of the pond for further treatment. So they have their finfish, so to have this further treatment of the water before the water is used in the pond culture system. And so far, they haven't encountered any disease problem when they have started implementing this system in their pond. And of course, this is what Ms. Vin presented on nanobubble, the use of nanobubble for novel delivery of fish vaccines. So I will not talk about more on this because this had been presented, but the oxygen nanobubble is a novel delivery system for improving immersion vaccination in fish, offering a cost-effective, user-friendly vaccination strategy for farmers. But again, one of the issues here is, do we have any vaccine to use? We have the technology, but we need vaccines. I think I'll stop here. So if you have some questions, as I said, if I cannot answer some of them, I will call on Dongha and Kalaya. I don't know where Kalaya is. Thank you very much.
Thank you very much for the detailed information about the new technologies. We may have one question.
Thank you very much, sir, for your nice presentation. Basically, my question is on that bacteriophage, that lies V, that what I see from the presentation, that it is a broad spectrum phage, cocktails of broad spectrum phage. Can you throw more light on that? Because it is not working against one species, but several species coming under the different clades. So it is extremely broad spectrum. Can you throw some light on that?
Sure. So according to the Dr. Ketia, the CEO of Unifas, they have this a bank of phages on different Vibrio pathogens, and depending on what is your target pathogen in your pond system, then they can develop this concoction of phages for effective treatment or of that particular pathogen. So if you have several pathogen Vibrio species in your pond that you want to target, so you inform them, they will develop this phage concoction from their stocks of different phages for Vibrios, and this is what you will apply in your ponds. So phages are very, very specific. So if this is intended for Parahemolyticus, they will only attack Vibrio parahemolyticus. The other bacteria in your pond, it will not be affected. So it has no issues on, unlike chemicals, when you apply it on a pond, it will affect all the bacteria there. It will kill all the bacteria. But phages will be very, very selective depending on the pathogen that you are targeting.
Sir, is it a farm-specific cocktail preparation, or it is a broad spectrum which is intended for designed use at any farm?
Sorry,
No, like if suppose a farm is getting some kind of pathogens, so are you selecting based upon the pathogen preparing the cocktails, or cocktail is already there which can work in any of the farm based upon whatever outbreaks comes?
Sorry, I did not get it. As I said, it depends on your target pathogen, okay? So this lyse V can be, the concoction, the composition of the lyse V can be changed depending on the request of the farmers. Yeah.
Okay, just I wanted to have, what is the logic behind mixing phage with the immunostimulant? Phage and immunostimulant, there is one.
Oh.
What is the logic of combining them?
Kalaya, where are you?
Yeah, I don't think it makes any sense.
No. The reason behind that is they are trying to target both the pathogen and at the same time increase the resistance of the host or the shrimp or fish. So this is a double, it is like using immunostimulant and antibiotics at the same time. So the phages will attack the pathogen while the stimulants will increase, will boost the immune system of the host. I think this is related to the regulatory problem because what they want to present is to present it as a feed additive, and immunostimulants are feed additive. So they will just add the phages to make it, yeah. Because if it is a treatment, if it will be presented as a treatment product, then it will undergo a different regulatory process and it will take time, just like the vaccines. So vaccines, we don't have very few, if we have, commercially available vaccines, and it cannot reach the farm level because of these regulations in vaccine registration. So they are trying to avoid that regulatory issues. I hope I have the same thing with Kalaya. Yes, one question. Oh, yeah, okay.
Very, very informative session, sir. And I have a question. What is the...
Can you...
Yeah. What is the impact of beneficial Vibrios? What is the impact of lyse V on beneficial Vibrios?
As I said, lyse V, these are bacteriophages, and these bacteriophages are very specific to the pathogen. So the beneficial Vibrios, if you have there, will not be affected, or any other bacteria present there will not be affected. So they will only target, if you are targeting Parahemolyticus, they will only target Parahemolyticus. If you have another pathogen there, Harvey, for example, but your phage concoction is only for Parahemolyticus, it will not affect the Harvey.
Is it stain specific also? Non-virulent stains also got affected by this Vibrio?
Okay, can you repeat that?
Non-virulent stains also get affected by this phage? If it is Vibrio specific, the two species specific, Vibrio paramelecticus is getting affected. Whether it is stain specific also?
Yeah. Some phages are very strain specific because this one, the Parahemolyticus that they have developed, is for append. So the one Parahemolyticus that are producing the peer A and peer B toxic genes. So the other Parahemolyticus might not be affected by the phage. So very, very specific. Phages are very, very specific.
Thank you.
Just like viruses also. So some of the viruses, of course, except white spot, some viruses are very specific in their host.
Yeah, just to add to your comment, to your question as well, that we have already started using phages for Moritella in the field in Norway, and some of them are so specific, they're not even working towards the variants of the bacteria. So need to actually have a cocktail of several phages for several variants of the pathogen.
And that's what is lyse V. It's a cocktail of phages for, Vibrio species in general. But you have to exclude, of course, the beneficial Vibrio. So,
So it's a quick question. I'm just curious that how cost effective is nano bubble technology compared with the traditional aeration or the ozone system in terms of the energy and operational costs?
Yeah, the traditional aeration system, they just produce the bubble. As I showed in the first slide, this bubble will just go at the surface and disperse immediately. So nanobubble can stay in the water column for several days. So it can produce more, if you are using oxygen nanobubble, it can provide more dissolved oxygen for a longer time than the ordinary aeration system that are producing these ordinary bubbles, which can easily disappear.
So let's say that it's cheaper than the traditional or
No,
ozone
it's
system.
expensive. Okay. But the benefit that you can get from that, because it's not only for aeration, it's also used for water disinfection, just like what I showed you in the shrimp farm. So the nanobubble has more usage than the ordinary aeration. So for disinfection, some are for, of course, if we have vaccine, it can be used for vaccine delivery. So there is a lot of potential for the nanobubble technology. And another one working on the nanovaccineer is Chanarong. Where's Chanarong? Dr. Rodkum from Chulalongkorn.
Sir, I have a small question. Since we're talking about alternatives to antimicrobial resistance here, bacteriophage is being used even in the medical field. They have come up with resistance in bacteriophage being used as a therapeutic as well. So what do you think the timeline is before we get concerned about that problem here?
I don't think so, because AMR will develop if you are presenting, treatment or chemicals at a sub-lethal level. So that's where AMR is developing. So prophylactic use or misuse of antibiotics and antimicrobials, this will lead. So if you expose your pathogens to lower concentrations of these chemicals, then that's where resistance will develop. So they will get used to the presence of these chemicals. For phages, it's different because phages will automatically infect and kill these bacterial cells, so they don't have time to develop a resistance from it. Yeah.
Okay, thank you everyone. It's great to see so much discussion, but it is actually coffee time now, so I would encourage you to continue these discussions in the coffee break. Thank you.